Welcome back, everyone. We've got another really interesting batch of papers to delve into this week.
Yeah, some really fascinating stuff.
All fresh from longevitypapers.com.
Absolutely.
And as always, our mission today is to kind of sift through all of this incredible research
Right.
and pull out the most impactful findings, especially those that might challenge or,
Yeah.
make us rethink how we currently understand the aging process.
Absolutely.
Really looking for those game changers that might,
Right.
steer the field of longevity biotech in new and exciting directions.
Yeah, and a lot of this comes from credible institutions too, so that makes it extra exciting.
Exactly.
These aren't just, small preliminary studies.
Right.
We're talking about some really robust research here.
Yeah, so we've got a handful of papers to unpack today.
Yeah, 5 to be precise.
5, okay, great.
All published between Monday and Wednesday this week.
This week, okay, so hot off to the presses.
Fresh out of the oven.
Wow, okay. Why don't we jump right into it?
Yeah, let's get started.
And this first one that caught my eye comes from Christina Masbargs and her team at the Freshage Research Group
Okay.
over at the University of Valencia in Spain.
Interesting.
And it was published just this Wednesday on PubMed.
This Wednesday, okay.
March 19th. The title is,
Let me guess. Something about frailty.
It is about frailty.
I knew it. It's always about frailty.
This is a hot topic.
It is, for good reason.
Yeah. BCL-XL overexpression in T cells preserves muscle mitochondrial structure and function
Wow.
and prevents frailty in old mice.
That's a mouthful.
It is a mouthful.
But a very interesting mouthful.
Yeah, so, for anyone out there working on age-related frailty,
Yeah,
this is definitely one to take a close look at.
absolutely. Anything that hints at preventing frailty is huge.
Huge news.
Huge news.
So what's really groundbreaking here to me isn't just that, BCL-XL is involved in aging.
Right.
But it's the direct link
Yeah.
this research uncovers between the health of T cells, which are part of the immune system, of course. And the physical frailty of muscles.
You don't often think about those two being connected.
Not immediately, right.
Right.
It suggests that we really need to start thinking about aging
Yeah,
in a much more interconnected way,
absolutely.
where bolstering one system, like the immune system,
Right.
could have these ripple effects on other seemingly unrelated systems like our muscles.
It's all connected.
Yeah.
So,
So how did they actually go about doing this?
so they found that by increasing the levels of this anti-apoptotic protein
Okay.
called BCL-XL.
I've heard of that one.
You've heard of that one, yeah.
It's like a survival switch, right?
Yeah, think of it as a survival switch for these T cells.
Keeps them from dying.
It prevents them from dying prematurely.
Interesting.
And they specifically targeted T cells in aging mice.
Okay.
And what they saw was this cascade of positive effects.
Like what?
Yeah, so let's delve into those.
What specific benefits did they observe that make this research so potentially impactful?
Well, first of all, the mitochondria in the muscle tissue
Okay.
of the mice with the boosted BCL-XL levels,
Right.
they maintain their structure and function much better with age.
Ah, so healthier mitochondria,
Exactly.
which are essential for cellular energy.
Absolutely. They're the powerhouses of the cell, and we know their decline is a key driver of aging.
Yeah, absolutely.
And this was specific to the muscle tissue, which is particularly relevant for frailty.
So it's targeting that.
Yeah, it's not just a general improvement, it's a targeted improvement in the system most affected by frailty.
That's really interesting.
And there's more.
Okay.
These mice, they actually showed a significant reduction in frailty.
How did they measure that?
Well, they had better grip strength,
Okay.
improved motor coordination,
Wow.
and even greater exercise endurance.
So like physically less frail.
Exactly. They were stronger, more coordinated, and had more stamina.
That's a big deal.
It is, because
Yeah.
it translates directly to a better quality of life as we age.
Right. If you can move better and you have more energy.
Exactly. You're more independent. You can enjoy life more.
It's huge.
And the researchers, they actually developed their own Valencia frailty score
Oh, wow. Okay.
to quantify all of this.
Interesting.
And the mice, with the increased BCL-XL, they consistently scored better as they got older.
That's pretty compelling.
It is not just an incremental improvement. It's a real, tangible difference in their physical capabilities.
Yeah, and the paper also mentioned something about upregulated BCL-XL in centenarians.
Right, which is really interesting.
Yeah, what do you think that means?
Well, it suggests that this pathway might be naturally upregulated in people who live exceptionally long lives.
So there's something there.
Yeah, it's a clue that this pathway might be playing a role in their longevity.
Okay, so we've got healthier mitochondria, we've got improved physical function.
Yep.
What else did they observe?
Well, they also looked at the role of T regulatory cells, or TREGs, which are a type of immune cell known for dampening inflammation and promoting tissue repair.
Okay, yeah.
And they found that there was an increased infiltration of these TREGs into the skeletal muscle of the mice with the BCL-XL boost.
So more of these good guys in the muscles.
Exactly. And the thinking is that this might be contributing to the improved muscle health and reduced frailty.
So it's not just the BCL-XL directly.
Right. It seems to be triggering a sort of chain reaction in the immune system.
Leading to a healthier environment in the muscle tissue.
Precisely. It's like a domino effect.
So it's a multifaceted approach, it sounds like.
It is, and that's what makes it so exciting.
But of course, we have to be cautious about extrapolating from mouse studies to humans.
Absolutely.
What are some of the skeptical considerations?
Well, first and foremost, mice aren't humans.
Right.
Their aging process is similar, but not identical to ours.
So we can't just assume it'll translate perfectly.
Right. We need human studies to confirm these findings.
What else?
Well, this study focused on overexpressing a single gene, BCL-XL.
Right.
And aging is incredibly complex.
It is, yeah.
It involves countless genes and pathways interacting.
So it's not just one thing.
Right. So simply boosting BCL-XL alone might not be enough to achieve the same effects in humans.
Okay. And you mentioned inflammation earlier.
Right.
Is there a potential downside to suppressing it too much?
Well, that's another important consideration.
Okay.
While chronic inflammation is definitely harmful,
Right.
some level of inflammation is actually necessary for healthy immune responses and tissue repair.
So it's a balancing act.
Exactly. We need to understand the long-term consequences of dampening inflammation through this mechanism.
Makes sense.
And finally, as with any initial study, replication is key.
Right.
We need larger independent studies to confirm these findings before we get too carried away.
So it's exciting, but more research is needed.
Absolutely, but it's definitely a promising avenue for future research.
Okay, so if you were in the lab right now, what kind of follow-up experiments would you be prioritizing based on these findings?
Well, I would definitely start by looking at human T cells.
Okay.
Especially comparing those from different age groups.
Interesting.
Including centenarians.
To see if there's a similar pattern?
Yeah, to see if BCL-XL levels and their impact on mitochondrial function show similar patterns in humans.
That makes sense.
And I would also explore using gene editing tools like CRISPR-Cas9.
Oh, wow. Okay.
To more precisely modulate BCL-XLA levels in human cells.
So you can really fine-tune it.
Exactly. That would give us much finer control and more direct insights into the mechanisms involved.
And would you look at other tissues besides muscles?
Absolutely. We'd want to investigate the broader impact of BCL-XL overexpression in T cells on other organs to get a better understanding of any potential off-target effects.
That's really important.
It is. We need to know if there are any unintended consequences.
And finally, would you consider combining this approach with other therapies?
Definitely. Exploring combination therapies that target both immune cell survival and muscle-specific aging pathways could be really powerful.
Yeah, so you're hitting it from multiple angles.
Exactly. That's often the most effective way to address complex problems like aging.
Okay, great. So that was a deep dive into the first paper.
Yeah, a lot to think about there.
Definitely.
A lot of potential.
So let's move on to the next one.
Okay.
Which also deals with targeting aging at the cellular level.
I like where this is going.
This one comes from Sandra Atlante and her team at the Istituti Clinici Scientifici Mogeri, IRCCS.
Okay, where's that?
It's in Italy.
Italy.
It was published in Aging Cell on Tuesday, March 18th.
Okay, just a day before the last one.
Yes, very recent.
It's very fresh.
And the title is A Xanthine Derivative with Novel Heat Shark Protein 90 Alpha Inhibitory and Senolytic Properties.
Wow, that's quite a mouthful.
It is.
Xanthine derivatives. Now, those sound familiar.
They do, right.
Isn't caffeine a xanthine derivative?
It is.
Interesting.
So that's one of the interesting things about this paper.
Okay, tell me more.
These researchers discovered a new xanthine derivative. They called it K5.
K5,
through a screening process that was specifically looking for compounds that could kill senescent cells.
So we're talking senolytics here.
Yes, this is a senolytic paper.
Exciting stuff.
So what makes this potentially a true step forward is that K5 not only acts as a senolytic, but it also inhibits heat shock protein 90 alpha.
HSP90 for sure.
HSP90, exactly.
So it's a twofer.
It's a twofer.
I like it.
And they even found that K5 extended lifespan in fruit flies.
Wow.
And reduced senescence markers in mice.
So it's showing promise in multiple model organisms.
It is, and that's always a good sign.
And the fact that it's a relatively well-tolerated chemical class is a big plus.
Yeah, it's not something totally new and crazy.
Right. We have some experience with xanthines.
So that's encouraging.
It is. It suggests it might be easier to translate into a safe and effective therapy.
Okay, so we've got this dual action.
Yeah.
It's not just getting rid of senescent cells.
Right.
It's also messing with this HSP90 protein.
So why is that a good thing?
Yeah. Why target HSP90 in the context of senescence?
Well, HSP90 is a chaperone protein.
Okay.
It helps other proteins fold correctly and stay stable.
So it's like a quality control manager?
Exactly. It makes sure the proteins are doing their jobs properly.
Okay.
And senescent cells. They often rely heavily on certain chaperone proteins.
Including HSP90.
Including HSP90.
To survive all the stresses they're under.
Right. They're like, help me, HSP90, I'm falling apart.
So by inhibiting HSP90,
yeah,
you're essentially cutting off their lifeline.
Exactly. You're making them more vulnerable.
And that can lead to their death.
Right, through a process called methiosis.
Okay, so it's a different mechanism than some other senolytics.
It is. Many current senolytics target anti-apoptotic pathways.
Right. They prevent cells from committing suicide.
Exactly. But this is more like disrupting their support system.
And that makes them more likely to die naturally.
Precisely. It's a more subtle approach.
Very interesting. So the paper mentions that they tested K5 in vitro.
Yeah.
On different types of senescent cells, like fibroblasts and even some cancer cells.
Did it work? It did. It showed significant senolytic activity across various cell types.
So it's not just specific to one type of senescent cell.
Right. It seemed to have broad activity.
That's really promising.
It is. It suggests it could be effective against a wider range of age-related conditions.
So we've got the in vitro data.
Yeah.
What about in vivo?
Well, they tested it in fruit flies, and they saw a significant lifespan extension.
That's always exciting.
It is. It's a good indicator that it might have potential for extending lifespan in other organisms as well.
But fruit flies are fruit flies.
Of course.
What did they see in the mice?
In the older mice, they observed a reduction in several markers of cellular senescence.
So it's actually clearing out those senescent cells.
Or at least reducing their burden.
In a living organism.
In a living organism.
That's a crucial step.
It is. It's moving from the petri dish to a more complex system.
And that's what we really need to see.
Right, to assess its translational potential.
Okay, so you mentioned earlier that K5 seems to be quite selective for senescent cells.
Yeah.
They reported a high senolytic index.
Right.
Can you explain what that means?
Sure. So the senolytic index is essentially a measure of how good a compound is at killing senescent cells without harming healthy cells.
Okay, so it's all about selectivity.
Absolutely. We want to target the bad guys without hurting the good guys.
Right. That makes sense.
So the higher the senolytic index, the more selective the compound is.
And what was the index for K5?
They reported an index greater than 1320 for one cell type.
Wow.
And greater than 770 for another.
So that's really high.
It is. It suggests that K5 is much more toxic to senescent cells than to healthy cells.
And that's exactly what we want.
It is. It minimizes potential side effects and maximizes the therapeutic window.
Okay, so K5 is a xanthine derivative.
Yes.
And we know that some other xanthines, like caffeine, are relatively safe.
Relatively safe, yeah.
Does that suggest that K5 might also have a good safety profile?
Well, it's certainly a possibility.
Okay.
The structural similarity to known and relatively well-tolerated compounds is encouraging.
So it's a good starting point.
It is, but of course we need to be cautious.
Right.
We can't assume that just because it's a xanthine, it's automatically safe.
So more research is needed.
Absolutely. Extensive preclinical testing is essential.
Including long-term toxicology studies?
Yes, to fully evaluate its safety and tolerability.
And it's definitely a promising lead.
It is. The dual action mechanism, the high senolytic index, and the potential for good tolerability make K5 a really exciting compound.
Definitely one to keep our eyes on.
For sure.
All right, let's move on to the next paper.
Okay.
Which takes us into the world of neuronal aging.
Ooh, the brain.
The brain, yes.
Always a fascinating topic.
It is.
Very complex.
This one comes from Wayne Mitchell and colleagues at Brigham and Women's Hospital, Harvard Medical School.
Harvard.
Published in Aging Cell on Wednesday, March 19th.
Same day as the first one.
Yep, busy day for aging research.
It seems so.
The title is a bit of a mouthful. Ready for this?
Hit me.
Intricate integration of chronic neuronal mitochondrial stress into neurotransmission processes via TMBM2-dependent K2 plus equilibrium, driving metabolic adaptation and behavioral changes for the regulation of aging.
That's a lot to unpack.
It is, but the implications for neurodegenerative research could be huge.
I'm intrigued. Let's break it down.
Okay, so this research was done in C. elegans.
C. elegans, those tiny worms.
The tiny worms, yes.
They're surprisingly useful for aging research.
They are. They're a great model organism.
Simple but powerful.
And what they found is that a protein called TMBIM2
TMBIM2
plays a vital role in how neurons cope with long-term mitochondrial stress.
So we're back to mitochondria.
We're back to mitochondria. They're everywhere.
Seems like they're at the heart of everything aging.
It does seem that way.
So what does this TMBM2 protein do?
It helps maintain calcium balance within the neurons.
Calcium balance. Why is that important?
Well, calcium is like the signaling molecule of the nervous system.
Yeah, okay.
It's involved in everything from neurotransmitter release to neuronal excitability.
So too much or too little calcium can really mess things up.
Exactly. It's all about balance.
So TMBIM2 is like a regulator.
It's like a thermostat for calcium,
keeping everything at the right level.
even when the mitochondria are under stress.
So it's helping the neurons adapt.
Exactly. It's a protective mechanism.
That's really interesting.
And they found that this calcium regulation had a ripple effect on the worm's metabolism and even its behavior.
And ultimately on its lifespan.
Exactly.
So it's connecting mitochondrial stress in neurons to the overall aging process of the organism.
It's a pretty profound finding.
It is. It highlights the interconnectedness of these systems.
And it suggests that protecting neurons from mitochondrial stress could have far-reaching benefits.
Beyond just the nervous system.
Exactly.
So what are the implications for us humans?
Well, we're obviously more complex than worms.
Much more complex.
But the underlying principle that neurons have these sophisticated mechanisms to deal with mitochondrial stress.
Right.
And that these responses can impact aging.
That's likely to be conserved.
So it's worth investigating.
Absolutely. We need to find the human equivalent of TMBIM2 and see if it plays a similar role.
And if it does,
then it could be a really promising therapeutic target
for preventing or slowing down neurodegenerative decline.
Exactly. Imagine if we could boost the activity of this protein
or its downstream pathways
to help our neurons better cope with mitochondrial stress.
That could be a game changer.
It could be a way to preserve cognitive function and extend healthy lifespans.
So a lot of potential there.
Huge potential.
Okay, let's move on to our final paper for today.
All right.
This one is all about measuring biological age.
Ooh, epigenetic clocks.
Epigenetic clocks, you got it.
I love this stuff.
It's a fascinating field.
It is. It's like looking into the future.
This study was published in Geroscience on Monday, March 17th.
Okay.
It comes from Angelico Mendy and Tesfaye B. Mersha at the University of Cincinnati College of Medicine.
Cincinnati.
And the title is Epigenetic Age Acceleration and Mortality Risk Prediction in U.S. Adults.
So they're looking at how well these clocks can predict who's going to die sooner.
That's essentially it.
And how does this study stand out?
Well, they didn't just look at 1 or 2 clocks. They examined 9 different ones.
Wow. 9.
Including the well-known Horvath clock and Hannum clock.
Those are the classics.
They're the classics.
What else?
They also included some of the newer, more advanced clocks. Like PhenoAge and Grimmage.
Grimmage, that one's getting a lot of buzz.
It is. It's considered one of the most accurate.
Interesting.
And they even threw in the Dundon-Pohm pace of aging measure.
So a really comprehensive comparison.
Exactly. They wanted to see which one was the best predictor of mortality risk.
And what did they find?
Well, Grimmage came out on top.
Grimmage. No surprise there.
It consistently outperformed the other clocks.
In predicting all-cause mortality.
As well as mortality from cardiovascular disease and cancer.
So the big killers.
The big killers, yes.
That's significant.
It is.
It means Grimmage might be capturing something really important about the aging process.
Yeah, something that's directly linked to disease and death.
So why is Grimmage so good?
Well, unlike some of the earlier clocks that were mainly focused on predicting chronological age, Grimmage incorporates information about physiological factors.
Like what?
Like blood markers, smoking history, things like that.
So it's not just looking at DNA methylation.
It's taking a more holistic view of aging.
That makes sense.
And it seems to be capturing more of the biological processes
Right.
that are actually driving aging and disease.
Not just the passage of time.
Exactly.
So it's a more clinically relevant measure.
It is, and that's what we're really looking for in a biomarker.
And what about PHENOAGE?
PHENOAGE also did well.
So it's not just grimmage.
Right. It seems like these newer clocks that incorporate more health-related data are really pushing the field forward.
They're giving us a more accurate picture of biological age.
Which is crucial for developing and testing interventions.
Absolutely. If we can track biological age more accurately, we can better assess the effectiveness of our therapies.
Okay, so the study also found some interesting differences based on race and ethnicity.
Okay.
Particularly among Hispanic participants.
What did they observe?
Well, some of the clocks, including Horvath, Hannum, and even Grimmage, were less predictive of mortality in Hispanic participants.
That's interesting.
Compared to non-Hispanic white participants.
So there are racial and ethnic differences.
It seems so.
In how these clocks perform.
And that raises some important questions.
Like what?
Well, about the generalizability of these clocks.
Right. Do they work equally well for everyone?
And it highlights the need for more diverse data.
Absolutely. We need to make sure these tools are accurate and applicable across all populations.
It's about inclusivity in science.
It is. We can't develop therapies that only work for certain groups.
So these epigenetic clocks are powerful.
They are.
But we need to keep these limitations in mind.
Right. They're still under development.
And we need more research.
More research is always needed.
To understand how they work.
And how to use them responsibly.
So as you can see, this week's research from longevitypapers.com has given us a lot to think about.
Definitely food for thought.
From new ways to target senescent cells, to the intricate connection between mitochondrial stress and neuronal health.
It's all connected.
And the ongoing progress in measuring biological age.
It's an exciting time to be in this field.
It really is. The pace of discovery is incredible.
And it's accelerating.
So we hope this deep dive has been helpful.
Yeah.
And has given you some new insights.
Hopefully some inspiration too.
What stood out to you?
What are you going to explore further?
What questions are you pondering?
We encourage you to dig deeper into these papers.
And think about how these findings might impact your own research.
And here's a final thought.
Okay.
Given the increasing accuracy of these epigenetic clocks, how can we best integrate them into our research pipelines?
To accelerate the development
of effective longevity interventions.
It's a challenge and an opportunity.
It is. Let's use these tools wisely.
To create a healthier future for everyone.
To a longer and healthier life.
To a longer and healthier life.
That's the goal.
That's the goal.
Thanks for joining us.
Thanks, everyone.